Lipid transport in the blood is difficult because we have already studied lipids are non-polar and blood is polar. We know carbohydrates are polar, so that is why it is easy to transport a carbohydrate. But how to transport lipid from one part to other part in the blood, because the blood is polar and lipids are non-polar. So body has made a strategy to travel or transport lipids, that is in form of lipoprotein. So these lipids are attached to a protein part, and therefore the outer covering contains protein, inside core contains lipid, and that is how a lipid can be transported. So inside is the lipid part, outside we have a protein part, and this protein part is known as apoprotein. So the lipid either can be triglyceride, which we have already studied, which contains 3 OH, that is a glycerol, and we have 3 fatty acids attached to it. So it is triacyl glycerol, which is also known as a triglyceride. Or it can be either transported in phospholipid, that means to a lipid part a phosphate group is attached. Or it can be transported in cholesterol form, or it can be transported in cholesterol ester form. Cholesterol is required for vitamin D synthesis, so cholesterol is also not bad. So triglyceride we have already covered in non-polar, non-polar and non-polar. So that makes it a complete triglyceride as non-polar. Phospholipid, lipid is non-polar and phosphate group is polar, so that makes it an amphipathic molecule. Now if you have a look at the structure of a cholesterol, alcohol plus fatty acid, that makes it a fat or a lipid. So alcohol is polar, fatty acid is non-polar, so that makes this as the non-polar. Fat is a non-polar. Cholesterol is a alcohol, that is amphipathic. So cholesterol is a amphipathic, and if it is attached to a non-polar fatty acid, that makes it a fatty acid. Is what, guys? Fatty acid is also polar, right? Fatty acid is a polar, but fat is non-polar, right? We have already studied this. Fatty acid is polar because it can make COO negative, that is a charged molecule. So cholesterol is an amphipathic molecule. It is an alcohol, but it is amphipathic molecule. Then a fatty acid is polar, and cholesterol ester makes it a non-polar. So if you see a lipoprotein, all the non-polar is always in the center. So we have non-polar in the center, such as triglyceride, or we have cholesterol ester. But if you see, the polar is towards the periphery, such as in phospholipid, phosphate is polar, which is towards the periphery, and phospholipid, lipid is non-polar, which is towards the core. Likewise, we also have cholesterol, OH, alcohol part is towards the periphery, and cholesterol towards the lipid part, towards the core. So diet, we eat fat. It is exogenous fat, which is in form of triglyceride. Triglyceride is what? A glycerol attached to 3 fatty acids. So this is the exogenous fat. This is the exogenous triglyceride. That means we are taking this fat from outside. It reaches to the intestine. Now it has to travel to the liver. It has to travel via the blood. We need to make this a lipoprotein so that it can transport. So up till now we have this triglyceride which reaches to the intestine. When it reaches to the intestine, triglyceride is surrounded by, it makes it a lipoprotein. It is surrounded by a protein part which is known as apoprotein. So this is ApoB48, and this molecule makes it a chylomicron. So a chylomicron is a lipoprotein which can further transport via the blood. So we have such kind of lipoproteins. What are the different kinds of lipoprotein via which the fat travels? Either it can be chylomicron. This chylomicron from intestine reaches to the liver, to the other tissues, and whatever is the left in this chylomicron is known as chylomicron remnant. So leftover, when it becomes, this molecule becomes small, it becomes a chylomicron remnant. Then we have VLDL. VLDL is synthesized by the liver, and it is the endogenous triglyceride. So basically what liver is synthesizing in form of fat is VLDL. So it is the endogenous triglyceride. The remnant of VLDL is known as IDL. Then we have LDL, and then we have HDL. So what is HDL? HDL is a empty core. Nothing is there in the core. Lipid is not there in the core. But we have protein present at the periphery. So this is protein A, C or E, ACE protein. So we have phospholipid, phosphate at the periphery, lipid at the inside in the phospholipid part. Also we have an enzyme that is present at the periphery, which is LCAT enzyme. So basically a core is empty in case of HDL. Now this empty core goes to the periphery, takes up the lipid, takes the lipid and goes to the liver again. So this is HDL, which is basically a empty core which takes up the lipid from periphery towards the liver, and this kind of transport is known as reverse cholesterol transport because it is taking from the periphery towards the liver. So if you see all these lipoproteins, which is chylomicron remnant, VLDL remnant, LDL and HDL, density is from least towards the maximum density. So very easy way to remember is density is inversely proportional to triglyceride content. As I told you, chylomicron has got the least density. That means chylomicron has got the maximum triglyceride content. And HDL has got the least triglyceride content. Now, if you can recall, I told you all this triglyceride, when it is attached to ApoB48, it makes it a chylomicron, but the HDL core is empty, right? So that makes it a least triglyceride content. Now density is also inversely proportional to the size of the lipoprotein. That means HDL, which has got the maximum density, it has got the least size. The lipoprotein size is least for HDL, and the lipoprotein size is maximum for the chylomicron. And this density is directly proportional to the percentage of protein. So HDL has got a maximum protein. It has got a maximum phospholipid. And basically the lecithin, which is the maximum in HDL, and this kind of questions can be asked in multiple choice question. So maximum proteins is present in HDL because HDL contains maximum density, and density is directly proportional to the protein. And since chylomicron has got the least density, it has got the least protein. So we know SCR are the cells that stores the lipid. RER are the cells that synthesize these are the organelle of the cell, that's RER synthesize the proteins. So talking about chylomicrons. Chylomicrons are the exogenous triglyceride, the fat that you are taking in the food. And so if you remember this formula, we talked about that if you are taking a carb source, 50% of that carb source gives energy. Rest 50% is stored, right? It is stored in the body. Now out of that 50% which is stored, 10% as the glycogen and rest 40% is saved as fat. Now the fat that, this is the carbs that you are taking, the fat that you are taking in the diet, that is a exogenous fat, and the fat that body is synthesizing from carbohydrate, it is the endogenous fat. The exogenous fat is chylomicrons. The endogenous fat, which body is synthesizing from the excess carbs, that is going to be VLDL. These are the bad fats. So exogenous triglyceride is coming from the intestine and going to the peripheral tissues. Lipid form of this is triglyceride and the protein is ApoB48. The remnant of this chylomicron remnant, the lipid is going to be triglyceride plus cholesterol. So everywhere when you see remnant, then there is cholesterol, and everywhere when you see remnant, the protein is going to be ApoE. So for remnant, RE, you can remember cholesterol, the lipid, and ApoE is the protein. So ApoB48 is the protein for chylomicron, and for remnant we add ApoE. Then VLDL, very low density lipoprotein, are the endogenous triglyceride, endogenous fat that body is producing from carbohydrate, and it is coming from the liver, which is forming this endogenous fat or endogenous triglyceride going to the peripheral tissues. So this is basically a form of triglyceride only, and the protein here is ApoB100. Then VLDL remnant, remember the formula for remnant. We add cholesterol for lipid and we add ApoE for the remnant in protein part. LDL, low density cholesterol, is going to peripheral tissues, and the form of lipid is cholesterol. Here it is not triglyceride, it is cholesterol, and the protein is ApoB100, which is for VLDL also, plus we have ApoE. Now till now we studied ApoE is for remnant, and LDL, further LDL and HDL have no remnants, therefore in both of these we add ApoE. So ApoE for LDL also, ApoE for HDL also. And HDL, which is a good fat from the peripheral tissues going to the liver for the oxidation. So it is a good fat, and the fat is cholesterol esters, and the protein is ApoAce, A, C and E, which is frequently asked. Triglyceride content is lowest in, as I told you, the good fat. Which is the good fat, guys? It is HDL, since it is going from the tissues, peripheral tissues, to the liver, so that the fat is broken down and to give off energy. So HDL is a good form of fat. Lipid, which accumulates in the fatty liver. So basically it is talking about the endogenous fat, and endogenous fat is triglycerides. Major lipoprotein of the blood, it is a LDL, which is a beta lipoprotein. ApoB100, if you remember, ApoB100 is a major lipoprotein for VLDL as well as for LDL. Apolipoprotein B has the highest predictive value in morbidity of coronary heart disease. Alpha and omega oxidation are the alternative pathway for the oxidation of fatty acids, in which no initial activation is required and no energy is required. Remember, alpha oxidation of fatty acid requires no ATP. And monooxygenase are required. So dash form of chylomicron is used to transport the cholesterol from intestine to the liver, and if you remember, it is the HDL, which is, it is talking about the HDL. So from the intestine, it is taken, no sorry, that is taking from the peripheral tissues. So it is asking about the intestine to the liver. HDL takes from the peripheral tissues to the liver. So if you recall, intestine to the liver, which is the fat that is going from the intestine to the liver. So it is ApoB, which is taking, so that is intestine to the peripheral tissues, or these are the, that is the triglyceride, that is Apo. So this is our ApoB form of chylomicron, ApoB. Cholesterol is maximally carried in, it is carried in LDL form, and as we have discussed that, major lipoprotein in the blood, it is LDL, or we can say beta lipoprotein. Right, now dietary cholesterol is transported to extrahepatic tissues. So from the diet, that is from the intestine to the peripheral tissues. So it is going to be LDL. ApoA is present in, ApoA is present in HDL. Remember, in HDL we have studied A, C and E. A and C apoprotein are only seen in HDL. So ApoA is present in HDL. Chylomicron and VLDL remnants are metabolized with the help of ApoE. So remnants, remember the formula for remnant, it is ApoE is the protein. So dash is the major source of extracellular cholesterol, and it is LDL, low density lipoprotein. Most important lipoprotein in causing coronary artery disease. So the bad cholesterol, which is the bad cholesterol, it is again LDL, which is the bad cholesterol. Receptors present in the liver for uptake of LDL are ApoE and ApoB100. So for LDL we have B100, and ApoE is the remnant of LDL. So remember, the remnant we have ApoE. Apart from that, LDL and HDL, they have no remnants, therefore ApoE is present in LDL and HDL. Apart from that, all the remnants like remnant of VLDL, remnant of chylomicron. So you need to memorize that table. There are so many questions that they ask from this table. Many function of ApoC2 activity, the main function of apoprotein C2 increases, it is the increasing the activity of lipoprotein lipase. So ApoC1 activates the lipoprotein lipase, while on the other hand ApoA2 does the opposite, that is inactivation of the lipoprotein lipase. Now, LCAT gene makes the enzyme LCAT, which is lecithin cholesterol acyl transferase, and this enzyme plays important role in removing the cholesterol from the blood to the tissues. So, activator of LCAT, it is APO1, whereas APOA2. APOA1 is the activator, APOA2 inhibits the LCAT activity, and the purpose of function of this LCAT, which is lecithin cholesterol acyl transferase, is it takes the cholesterol, it removes the cholesterol from blood to the tissues. Oligodendrocytes helps in expression of the brain lipid binding protein. Now coming to the diseases related to lipidemia, so increase in the hyperlipoproteinemias. So basically there is going to be increase in the lipoproteins in the body. Which lipoprotein is increased? Based on that we have a classification of disease. So type 1 is familial hyperchylomicronemia. As the name suggests, familial means it is a hereditary condition. Hyper means increase. Increase in what? Increase in chylomicrons. It is saying chylomicronemia. So chylomicrons are increased. So the triglyceride is increased in case of type 1 hypoproteinemia. Type 2 hypoproteinemia, it is familial hypercholesterolemia. So basically again familial disease, hyper means increase and cholesterol. So here the cholesterol is increased in case of type 2. So basically when I say cholesterol, that means we are talking about LDL. So LDL is increased here. So cholesterol is increased in type 2. Type 2A this is. Type 2B is familial combined hyperlipoproteinemia. So it is combined, first of all, combination of lipoproteinemias. That means we are talking about LDL plus VLDL. So both are VLDL. That means both are increased, VLDL and LDL. So this is cholesterol plus triglyceride, both are increased. Then we have type 3 in which dys-beta lipoproteinemia. This is a broad beta disease in which the remnant is increased. So type 1 in which chylomicrons are increased, that means triglyceride increase. Type 2 hypercholesterol, increase in the cholesterol, LDL increase in the cholesterol. Type 2B in which LDL and VLDL both are increased, so cholesterol and triglyceride are increased. Type 3 is the remnant increase. So enzyme deficient in type 1 hyperlipidemia or hyperlipoproteinemia, which is also known as chylomicron syndrome. It is lipoprotein lipase. The enzyme that is deficient in type 1, it is lipoprotein lipase. So here deficiency of lipoprotein lipase, which is chylomicron. Chylomicron syndrome, that means increase in the chylomicrons. Primary familial hypercholesterol, that means increase in the cholesterol. We are talking about type 2A, is due to deficiency of LDL receptor. So there is increase in the LDL in type 2A, which is hypercholesterol, increase in the cholesterol. Again, you need to remember guys this table very well, and apart from that, it is very important that you have to memorize this table also. So you need to practice this table in which chylomicron, we know chylomicron are the exogenous fats, and ApoB48, these are the triglyceride. VLDL, which is an endogenous fat, again triglyceride, and ApoB100. LDL, ApoB100 plus E, because there is no remnant in LDL, and the cholesterol that is going to the peripheral tissues. Then peripheral tissues to the liver, which is HDL. So HDL is bad, HDL is good, and LDL is bad, and LDL is found in the blood. So HDL increased is not indicative of hyperlipidemia type 2A. Apart from that, if you remember, type 2A is increase in the cholesterol, familial hypercholesterolemia, that is increase in the LDL. So increase in the LDL is seen in type 2A, not the increase in the HDL. Prediction type 2A hyperlipoproteinemia is due to excessive amount of. So excessive amount of LDL we already talked about. Type 2A is excessive amount of LDL, whereas in type 2B there is elevation of VLDL. Apart from that, LDL and also intermediate density lipoprotein is increased in case of type 2B. So if you have to recall, we will recall all these again. Type 2, first is type 1. Type 1 hyperproteinemia is increase in the chylomicrons. So we are talking about increase in the triglyceride, which is familial hyperchylomicronemia. This is chylo, not cholesterol. It is chylomicrons. Chylomicronemia. Then type 2A, it is increase in the cholesterol, then increase in the LDL, and this is familial hypercholesterolemia. Then type 2B. Type 2B is cholesterol plus triglyceride. Here was triglyceride increase, and LDL is increased. Apart from that, also VLDL is increased in case of type 2B. So it is familial combined hyperproteinemia, lipoproteinemia. And 3 is very easy. It is remnant, so dys-beta lipoproteinemia. Now again, let's see. It is type 2A, Fredrickson. Type 2A is increase in the LDL, while type 2B is increase in the VLDL, LDL and IDL, which is intermediate type. Now the best marker for dyslipidemia, it is the ratio which is LDL divided by HDL. HDL is the good fat, LDL is the bad fat. A young man on examination has found to increase the serum cholesterol on further study. Only LDL is increased in the cholesterol. Rest everything is normal. And if LDL is increased, that means we are talking about hypercholesterolemia, right? Then the history of same is seen in the fathers too. So there is defect in the LDL receptor. It is type 2A. Type 2B means increase in the LDL. Apart from that, increase in the VLDL and IDL, which is intermediate density, is also increased.